All right. So in the last lecture, we talked about the enzymes, enzymatic reactions, as well as catalysis, and some examples too, like the serine proteases and things like that. Today, we're going to keep discussing the enzymatic reactions, but focusing on the mechanisms. If I can move forward. Here we go. So today we're going to mostly focus on the catalytic mechanisms, how they actually react on the different steps of the enzymatic reactions and so on. So typically you put these chemicals or the compounds, or it can be proteins too, in the reactions that are taking place. And these are written with the as the chemical compounds here, and also we use these like curved arrows here. Basically shows the path of electron pairs. So it starts from the electrons where it originated from, and then how it forms the some kind of bonds, which means that that's electron pairs. So this is an example here where it shows the interaction between the reaction between amine and the aldehyde or the ketone, basically, which has this ketone group right here, and this amino group here, which has the electron pair already, attacks the carbon here, and arrow means that there is a movement of electron to form the electron pair, in which case, in this case, makes the single bond, covalent bond, as you can see here. And then it forms the double bonds and so on, for keeping showing the basically how the electron pair is formed, or how the electrons flow. And these arrows basically go from the electron sources to the electron deficient center, which attracts the electron pair, basically. So you're gonna see this a lot when I show you the compounds and the reactions for their diagrams and things like this. We'll see the curved arrows a lot, and that always means that there's electron flows there to form the electron pairs. So there are different types of catalyses. And we're going to basically talk about all these 5 different kinds of catalyses one by one, which are really important. And you should be able to distinguish between their differences and the particular catalysis type from the other types. So the first one is acid-base catalysis. Of course, consider the acid-base reactions. And second one is covalent analysis, where the covalent bonding is involved there. Excuse me. And the third one is metal ion catalysis. As it says, this catalysis involves the metal ions, basically, which are positively charged. And that's also the electric or electrostatic catalysis too. The fourth one is proximity orientation effect. And the fifth one is preferential binding to ES complex, which is a transition state. And it does not mean that the certain catalysis just uses single type of among these different types of catalysis. They typically actually use combined catalysis types or the mechanisms. For example, acid-base catalysis can also use proximity and orientation effects, as well as preferential binding of transition state or the ES state or the complex too. All right, so let's go over again, one by one. The first one is the acid-base catalysis. And this catalysis basically, makes the rate acceleration via partial proton donation by a catalysis. It's a proton donation, so giving up the proton. So that's the property of being acidic, right? So that is also called general acid catalysis, or it can be also a partial proton abstraction, in which case, I mean, it's basically taking off the proton from the other partner, so it's prone to have the proton recover the proton. So that's going to be the property of base. So that's a general base catalysis, and this general acid or general base catalysis can be modeled easily by looking at this keto to enol isomerization. So we talked about this, long time ago when we talked about tautomerization, basically, where it can be keto form or enol form. And this is a pretty slow process actually. So here's an example of which compound contains the acetyl group. As you can see, CH3CO group here. And when you look at this, the proton here, the hydrogen atom here, can actually ionize from this. So it leaves, and then of course there's gonna be one electron that is left over, right? So that's gonna be basically moved to here. So again, it shows the curved arrow here, meaning that the electron flow, and then forms the electron pair, which is a double bonding. So there's going to be a double bond between these two carbons, and there's another double bond. So there's going to be a resonance, basically, that the electron can flow here and there. Makes the partial negative charge for both ends for this carbon as well as this oxygen. And later on, so this hydrogen as a positively charged can just leave, and the essentially the same molecule or the other hydrogen cation that is floating in the solvent can come here and easily attack this negatively charged, partially negatively charged group here to form the enol form. So again, this is a pretty slow process. So you can accelerate this by using catalysts. You can both use either general acid or general base catalysts here. For the general acid catalysis, it uses acid, of course, right? So here's an acid, HA, A is a salt, and A, basically this is gonna be ionized, so this hydrogen cation will easily attack this oxygen, right? Which will deprive this one atom here. So again, this is going to be similar to this shape, the transition state from between the keto and enol, and it will induce to leave this proton on the bottom, so it's going to be ionized and then will leave. So you're going to see that as a leaving group here. After that, it will make the enol form because of this excess hydrogen cation. But also there's a salt, which is a the conjugate base that is left as a A minus. And this one will be recovered back to the acid formed by the addition of water molecule. So when you look at the keto and enol form, this acid, even if it was involved in the middle, now it'll recover to its original form. So it's basically used as an enzyme. It wasn't, involved in any kinds of reactions here. General base is working in a similar way, but in the opposite way. So instead of giving up the proton, it'll take up the proton on the bottom here, and then make it partially positive. And then this, of course, this is a base, so it has the prone to, take up the proton here, right? So ended up just having this hydrogen atom detached from the original keto form, and then we'll form this the base with the hydrogen combined here, and then later on it will have the similar transition state here for the partially negatively charged region for this oxygen. So the another hydrogen cation can come here and attack it to form the single bond, and this one basically will be recovered back to the basic form. So again, this general base can be recycled again and again to boost this reaction. So as you can question. Both of them are basically faster than the just original reaction without enzymes. So these are it depends on the basically, we talked about the rate, the reaction rate K last time, right? So it depends on the K value, whether this is a faster or slower process. But typically when the enzymes or the catalysts are involved, it's a faster process. And also depends on the concentration of the substrates too, right? When it reaches to the equilibrium, getting to the later stage of this reaction, then this reaction will basically slow down, as opposed to the initial stage when there are lots of substrates. Then this process will be much faster. Okay. So as you can imagine, this is an acid-base reaction, right? So it'll be affected by the pH of the solvent. Um, because it's all about protonation, right? Having the hydrogen cation or the proton or the leading or the losing the proton. And basically the amino acids have quite a bit of, the functional groups and that are affected by this protonation. And so that means the proteins will also be affected a lot in terms of this pH, and that includes the enzymatic reactions. So especially aspartic acid, glutamic acid, cysteine, tyrosine, histidine, lysine, arginine, and so on. All of these amino acids can be easily affected by the pH of the environment, right? Because they are acidic or basic, right? What about cysteine? Is cysteine basic or acidic? Can you recall, the amino acid properties? What is it? Exactly. So it has the sulfhydryl group, and it's not, neither acidic nor, basic. But it still can be affected by pH because it has a sulfhydryl group, which where the hydrogen cation, hydrogen atom can leave pretty well, as a form of hydronium or the hydrogen cation. So same thing applies for tyrosine too, which has the hydroxyl at the end of phenyl ring, right? So again, it can be affected by this protonation, where whenever you see the hydrogen, it can be easily affected by pH, basically. So all the enzymes are optimized or evolved in a way that it works the best at the physiological pH range, which is about 7. A great example of acid-base catalyst is RNase A. And there are different types of RNAs, RNase A, B, C, and so on. And RNAs are basically the enzymes. When we call the nomenclature of the enzymes in the last lecture. A's is for enzymes, and the something that is attached to the A's as a prefix would be the it's a substrate, right? So it actually cuts the RNA molecules. That's why it's called RNase. And when you look at the active cleft or the active site of this RNase, which is a pretty small and simple protein, it contains two histidines, which works as a acid-base catalysts. So when you look at their mechanisms or the working steps here, the first step would be here are the two histidines, histidine 12 and histidine 119. And these numbers represent the residues, their position, relative position on the polypeptide. The histidine 12 here first is basic, right? And this histidine is supposedly basic too, but it's attached with the proton already. So it's not going to be basic anymore, but more like it can give up the proton, right? So it can actually work as an acid. So let's just follow the steps of this catalytic reaction here. So first, histidine 12, which is basic, can attack the hydrogen atom here, which is attached part of the hydroxyl of 2' region of RNA, pentose. So as you know, RNA has OH on the 2', as opposed to DNA has just hydrogen here. And that's why RNA molecule is more susceptible to any kinds of degradation or kind of stress, because hydroxyl is basically quite reactive. And this RNA uses that reactive functional group here. So it deprives this H basically and make the covalent bonding here, it's transient, but still covalent bonding, um, working as a base. So as it says here, it abstracts the proton from 2' OH and promoting the nucleophilic attack of this phosphorus on the phosphorus from this um, O minus now, which was the hydroxyl group, because this is negatively charged now. So it wants to bind to or attack the something that can have lots of positive, charges, or at least can accept some negative charges. So it's called the nucleophiles or nucleophilic molecules or atoms. And we're going to talk more details about this nucleophilic atoms later on. But anyway, it attacks this phosphorus in the phosphoryl group, making this a single bond, transient bond. And then basically it cuts out this phosphate, the phosphodiester bond here. So you're going to have another half of this molecule that leaves. And then you're actually inserting using the water molecule here, introduce it here, which will, and I actually didn't explain this yet, but here's another one. So histidine 119, like I told you, will work as an acid now. So it'll provide the hydrogen to this already broken phosphate group or the phosphoryl group. So to make it as the here CH2O, and then we'll add the H, which is the original form for the nucleotide, right? So it's gonna left like in this form, and then now it'll be a basic. This histidine 119 will be basic now because it lost the proton. And this histidine 12, which gained the proton, will work as an acid now. So in the sort of the opposite way now. So the step 2 would be, so it has this sort of a phosphate ring or the bridge here. So histidine 119, which is located on the bottom, will deprive the um, hydrogen cation from the water molecule. So that's going to be hydroxyl, which is quite reactive and negatively charged. So it's going to again attack the nucleophilic phosphorus right here to break the bond between 2' and 3' here. So it's going to have the hydroxyl now. And then this histidine 119 will recover the hydrogen right here, which is original form. So it's gonna work as an acid now. And um, the histidine 12 that was attached with the hydrogen will lose hydrogen now and give it back to the 2' of this RNA molecule, and then we'll make this as a living group. And again, this histidine 12 is recovered to its original form, meaning that it ended up not really didn't involve in any of this reaction where DNA RNA molecule is like cleaved, and also can be recycled for the new molecule. Does that make sense to everybody? I talked a lot basically, but the process is quite simple. That one histidine 12 worked as the base. Histidine 119 worked as an acid, and step 2 it actually flips that kind of role. So the second type of catalysis is a covalent catalysis, where again covalent bonding is involved for this catalytic reaction, and this catalysis achieves the rate acceleration through the transient formation but covalent formation of the ES intermediate complex. And ES, as you know, is the transition state intermediate. So usually the nucleophilic group attacks an electrophilic group. And again, nucleophilic group is basically like the nucleus or proton. So it's going to be basically negatively charged groups that is trying to find the protons there. And electrophilic group is the opposite thing, where it likes to accept the electrons. So it's going to be positively charged, and try to find its partner that is negatively charged. And it forms the covalent bond during the catalysis. And this is an example of that covalent catalysis reaction, where the amino group is reacting with the ketone or the aldehyde to form this again single bond between the carbon and nitrogen atom. And here, nitrogen atom is a nucleophilic group because it has an excess electron there, and carbon is typically is positively charged and likes to get more electrons to be stabilized. Here's a double bonding, meaning that it's not saturated in terms of its valency yet, so it can actually have another breach to be more like stabilized in terms of its electrostatic property. So it'll behave as an electrophilic group, and then, basically there's an attack from the nucleophilic group to form this single bond. And single bond again will have becomes to the double bonding because of again there's a excess electrons there. So electron pair makes again to make the double bond make ended up having this shift base. It's called imine, and this shift base is important because it happens a lot in the catalytic reactions in the living system. There are lots of carbons and of course aldehyde and ketones too in the living organism, as well as nitrogen-based molecules too. So there are going to be a lot of these kinds of reactions to boost the any kinds of a breakage or adding some kind of functional groups to carbon-based compounds in the living system. Again, shift base is basically whatever you see, the double bonding between the nitrogen and carbon, and they basically have a different functional group attached, to them. So an example of this covalent catalysis is the carboxylation of the acetoacetate, which you can see its structure here. And this acetoacetates have the leaving group carbon dioxide as the leaving group. And they're going to be on a transition state where it becomes to the enolate, where it does not have carbon dioxide portion, basically. And then there's another hydrogen cation added to this molecule to become acetone. And this is a sort of a conventional way to generate acetone. Um, and this is a quite slow and inefficient process. As you can imagine, I mean, this negatively charged oxygen have to fold it back to form the carbon dioxide. And then basically there's a free electron that used to be forming the within this that pushed from this oxygen, right? So it flows back there and satisfy the sort of the total negativity there to form the single bond between carbon and oxygen, which is negatively charged now. And then this one falls back to form the double bonding between carbon and oxygen, just like the original form. And then again, there's electron deficiency basically that pushes back down to this CH2, which is supposed to used to have the double bonding. So there's another like one valency there. And then this hydrogen cation comes and satisfies that valency to form the methyl group. Does that make sense to everybody? It's a lot of sort of, chemistry and organic chemistry there. And hopefully you guys, understand how it, works, because this is more like a review kind of thing. If you don't understand, just let me know. Sure. Okay. What is it? Oh, yes. So, like I told you at the beginning, doesn't mean that because this is a covalent catalysis, it doesn't mean that it uses only that one, types of catalysis. So in here, actually, which I didn't explain, but I already explained at the beginning, where the acid and base is involved there. So it's going to be both covalent catalysis as well as acid-base catalysis. Oh yes, absolutely. There could be there too. It depends on what kind of reactions it's involved, but it doesn't have to be acid and base all the time. But it uses quite a bit of that. For example, metal ion is involved there to form the covalent bonding too, where there's nothing related, nothing to do with the acid-base reactions there. So it is completely possible. Not good for the enzymes. So, well, that's a good question. So is the covalent bonding between the substrate and enzyme a good thing or bad thing? What do you think? So the second one, we're talking about the covalent catalysis. Is that a good thing or bad thing for the enzymes? Anyone? So it can be a bad thing because it stabilizes the ES complex and make it to like keep it that way for too long. Is that any other person? I mean, any other thoughts? Is that a good thing or bad thing? If it is a bad thing, then why would we consider that as a type of catalysis? Maybe it's an inefficient catalysis. Any other thoughts? I'm sorry. It should be good because it's what is it? Oh, got you. Um, so it can be a good thing because I mean it can be solved during the process actually, because there could be a multiple steps involved there. So we talked about the good enzymes or more efficient enzymes where it has to have a strong interaction between when it makes the ES complex. And it has to have a relatively weaker interaction with the product or just the substrate itself, right? Does that make sense? So the good enzyme has to have the strongest affinity to the preference to become the ES complex. So covalent bond helps that actually, makes this ES complex stabilized, and there are different processes involved there for breaking that covalent bonding. So that is completely fine, but it depends actually on the what kind of enzymes you're using. The enzymes or the molecules that are involved for this covalent bonding should be easily form the covalent bonding and also break it too, right? And that depends on the property of the molecules that make some kind of dipole moments quite quickly to become the positively charged or negatively charged, so you can easily attack the nucleophilic, electrophilic regions or something like that. And then it can be easily actually becomes back to the non-polar kind of state. So it loses that kind of bonding or the interaction anymore. So it really depends. Um, Good question. So what do you think? I mean, we just talked about this, right? So covalent bonding has to be made between enzymes and substrates. What do you think here? Are there any covalent bonding involved between enzyme and substrate? Yes or no? No? No, because you don't see that, right? There are some bondings here and there forming and, but it's not the direct interaction between the enzyme and the substrate. It doesn't induce the ES complex. So it's not going to be the covalent bond catalysis. Any other questions? Because it can be quite confusing. So, all right. So we just talked about this and this one too. So this as a- acetoacetate that becomes the acetone, this reaction can be accelerated by the adding this primary amine or the something that has amino group working as the um, catalyst. So let's take a look at here how it processed basically and where the covalent bonding is involved. So again, this has to be the covalent bonding between the enzyme and the substrate to form the intermediate state pretty strongly, right? So here's a acetoacetate, and here's an catalyst, which is a primary amine, and that gets in, and since this is nucleophilic, it has the amino group that has excess you know, um, electrons. So it'll easily attack the carbon here. Besides, this carbon has with among this ketone group, so it has a double bonding, meaning that it's more susceptible to any kinds of nucleophilic attack. So it'll replace this oxygen here, and will form the covalent bonding now. And this is again enzyme. This is not enzyme, I'm sorry, catalyst. And this is a substrate, right? So there's an intermediate state right here that mimics the enolate here. So it leads to this intermediate state much more efficiently there. And since this one replace this O, and the leaving is a hydroxyl because there's another hydrogen from the primary amine that leaves along with the oxygen. So that means that's positively charged, right? Right here. So it'll make it easier to move this electron to up here. So the carbon dioxide is formed, making the double bonds here, and works as a leaving group here, and that will form this double bonding, as I explained for the original reaction here. And then in the same way, the proton comes in, and then will basically attack this nucleophilic center, which is a carbon here that has the double bonding, again, more susceptible to the nucleophilic attack. So it'll form the methyl group here. And then, of course, there's excess electron here that falls up to the more um, electrophilic group, which is an amine group right here. So this amino group will recover this positive charge now, and then will eventually, if you add the excess of hydroxyl again, so this is a reverse reaction of this essentially, then this hydroxyl will replace this primary amine now. So this is going to be a leaving group. Ended up having the hydroxyl is left there, and of course hydroxyl can be easily deprotonated to become excuse me, become this ketone group, to become the acetone. Okay. So we talked quite a lot about the nucleophilic groups, electrophilic groups, or the atoms and things like that. Let's discuss a little further about these. So nucleophiles are basically nucleophilic substances, or the atoms, and the electrophiles are the other way, electrophilic groups, or the atoms. And this nucleophilicity or electrophilicity is related to the basicity or the acidity of the given group. So nucleophilicity, meaning that we try to get the protons, right? Try to get the protons, or the losing the electrons is the same as basic property. Does that make sense? Based on the Bronsted-Lowry theories that we used quite a long time ago, actually, for the second lecture or so, to talk about the base and acid definitions. And nucleophiles that we already have seen make the covalent bond, but it's a transient bonding, right, to the electron deficient centers. And bases, again, because the bases or the basic compounds abstracts the protons or giving up the electrons. It's the same thing, right? Basically, having the positive charges there, having the excess electrons or so, giving up the electrons or accepting the protons. The examples of nucleophiles are here. So basically, these ROH, which is alcohol, or having the sulfhydryl group, or the amino groups, or the imidazole group, which is in the histidine side chain, all of them are gonna be nucleophiles once they lose hydrogen cation, because these are literally the acids, right? At least for these two. These are acids, but when they lose proton, they're gonna work as a conjugate base, right? So it's basic. So that's why they're gonna be nucleophilic. Okay? And the examples of electrophiles are something that is positively charged. So he wants to actually, electrophile wants to get the electrons or negative charges there to be more stabilized. So the protons like a hydrogen cation or the hydronium ions, or any kinds of metal ions which are positively charged basically, will try to get the recovery the electrons, right? So electrophiles, as well as we talked about carbon atom, which has double bonding, so it's not saturated in terms of its valency. It still have availability to make the other single bonding to the other atoms. So it'll be easily attacked by the nucleophile because this is electrophile, as well as cationic imine in the shift base we talked about. So here's again shift base having the double bond between the carbon and nitrogen, and this nitrogen is positively charged, so working as a base, so called as a shift base, and basically working as an electrophile. Okay. Yes. So you can actually take it that way too. So the question was that can you think that the nucleophiles as base? And the answer is yes. Answer is yes. Is it? Answer is yes. Because these are basically, take a look at this, electrophilic form. These are basically conjugate base of an acid. Can you see that? These are the acids, right? So you can take it as a base actually. Base typically try to recover the proton, right? So it's the same as getting some positive charges. Try to get the positive charges. So you can take it as a nucleophiles. Try to get to the positive charged ones, and then get any kinds of bondings or anything. Same thing applies to the acidic molecules too. Positively charged, so wants to get the electrons, or the interact with the negative charged molecules. Again, if you are a little confused, or the like, if you want to sort of brush your memories, you want to have a refresher on the Bronsted-Lowry acid-base definition, or going back to the organic chemistry basically for this. Or you can always ask me the question, any questions you have. The third type of the catalysis is metal ion catalysis, and this is happening a lot. So it's widespread because almost 1/3 of all enzymes in the living system require metal ions for catalysis. And that's because just forming the amino, enzymes with amino acids have limitations in terms of their specificities or type of reactions and so on. So we talked about the chemical teeth, basically, right? That gives a lot of different kinds of functionalities of the enzymes. And since you have lots of different kinds of metal ions, you can actually recruit them and have a specific properties, or use their special properties for achieving the, basically the acceleration of the reactions. So there are two classes of the metal ion catalysis, depending on the strength of the interactions. If it is really tight binding of the metal ions to either enzymes or the substrate, they're going to be called metalloenzymes. And typically, the transition metals, the sort of resides in the center of the periodic table, are used for this forming the metalloenzymes, like irons, copper, zinc, manganese, and so on, cobalt. Um, Another class is the metal activated enzyme that have loose binding between the metal ion and the enzymes or the substrates. And typically, alkaline or alkaline earth metals are involved there, like sodium, potassium, magnesium, and so on, calcium. And they basically act in three different ways. The first way is that the metal ions bind to substrates and orient them properly for reactions. So it's not like they're really involved in any kinds of catalytic reactions, but only because it has a specific charges and preference to their partners, they're going to orient their substrate based on the electrostatic property. And the second way is that they mediate the redox reaction, which is oxidation reduction reaction. So where the transfer of oxygen molecule is involved during the reaction. So we're going to talk about this redox reactions much later, but basically this is about oxidation. But it's involved there, and of course, lots of metals can be oxidized pretty well, right? It's pretty reactive to the oxygen. So we'll have the transiently changing the redox state, and will induce the catalytic reaction there. And the last way is to stabilize or shield negative charges because they're positively charged. To stabilize the any kinds of enzyme um, molecule or the enzyme substrate the interactions in the active sites that is occurring. So an example of particularly this third one, stabilizing the negative charges, is the ATP that transferring the one phosphoryl group to glucose to form the 6-phosphate glucose 6-phosphate. And where here the substrate is the actual substrate is not just ATP, but it's a magnesium ion and ATP, because magnesium ion is always required for this reaction. And this magnesium basically shields or stabilizes these negative charges of oxygens right here that are attached to phosphorus. And this phosphorus, especially for the third one, is attacked by this hydroxyl of the exocyclic carbon of the glucose to form the covalent bonding there. So in order for this oxygen to attack the nucleophilic um, center, which is a phosphorus, it has to be relatively more positively charged, right? But since it is already surrounded by this negative charges, it'll be already stabilized, and so it's not going to be as reactive as it, um, without the oxygens around. So these oxygen negative charges are stabilized by magnesium ions, cations. Um, then this phosphorus will be a little bit more positive. Does that make sense? So it can be attacked by the by this nucleophile. All right. Here's another example of the metal ion catalysis, which is a carbonic anhydrase. And carbonic anhydrase gets rid of water molecule from the carbonic. I don't know why, but its volume is kind of fluctuating. So, but anyway, this enzyme, carbonic anhydrase, gets rid of water molecule from carbonic acid. So the reaction, when you just take into account of its name, the reaction has to go reverse, right? But basically it's just a boosting the reaction in both ways. That's what enzymes do, right? So it just was named that way. So basically, it boosts the reaction where the carbon dioxide and water molecules react to each other to form the carbonic acid. And in here, this carbonic anhydrase requires a zinc ion, which is sitting right in the middle of active site. And that is important to pulling all these 3 histidine residues to open the active sites, as well as serve as the stabilizing the a functional group that is critical for this reaction. So here's a carbon dioxide, and again, zinc ion are basically holding these 3 histidines, and histidines have imidazole on their side chains. So it's written as IM here, imidazoles. So here's the zinc, and since it's positively charged, it can get this hydroxyl group, which is negatively charged, and form the transient covalent bonding. So it can be considered as a covalent catalysis too at this point. And then since this is stabilized here and also oriented in the perfect way where it can interact with the carbon dioxide molecule in the active site, this hydroxyl attacks the nucleophilic center, which is uh, nucleophile attack to attacking the electrophile center, right? Which is carbon, and then forms the single bond between this hydroxyl and carbon dioxide to form the carbonic acid. And then it'll leave in the addition of water molecules, and then this enzyme, especially the zinc, will recover the hydroxyl. And again, there's going to be another hydrogen cation that will leave there. So it'll recover to the original form here, and then will be recycled for forming another carbonic acid. And the fourth type, so among 5 different types, the fourth one is proximity and orientation effects. And again, this can be happened along with the other types of catalysis. And this is basically about the property where the binding of enzyme um, like forces the substrate molecules oriented in a certain way, especially on the active site. So if there's not much of a binding there, then the substrate will just flow and kind of twist, or not really twisting, but the kind of turning, or the oscillating and things like that. It's going to move in every directions, which can lower the probability of the reaction that goes forward, because it has to meet the right enzyme or the other substrate for the reaction. However, if the enzyme can hold the substrate in the certain way really strongly, then it can induce the interaction between 2 molecules. One example here is the SN2 reaction, and hopefully you guys know what this reaction is, which was supposedly covered during the organic chemistry, or the even general chemistry too. This is the reaction where basically 2 molecules sort of exchanging their bonding, right? So there's A, B, and C molecules. There's interaction, the covalent bonding between A and B, but now it makes the B and C bonding, and A is the leaving group. Then it's going to be called SN2 reaction, and that typically happens with this backside attack. So it's not using the same kind of bonding regions, but it's going to be using the opposite side here. So there was an original single bond here between the 2 atoms. And then here's another one comes. So you can take these as a substrates. And then there's going to be a transition state where both have the partial bonding there. And then eventually the original bonding will be broken, and the new bonding will be formed like that. And in here, the orientation is really important, right? Because what if this green blob comes on the side, right? Like here, then it won't be able to comes to this region where it properly has the single bond. So the orientation is important for this backside attack, and these enzymes can definitely help that orient those substrate in the proper way. And also, I talked about this, the rotational and transitional motions can, cease there. So again, helps the substrates meet together, or some kind of functional groups come together for any kinds of reactions. And this is a kind of bad thing when you talk about, when you think about the entropy, because the molecule wants to just move everywhere, flow everywhere, right? But it's basically restrained inside the active site of the enzyme. So it'll lose the entropy, meaning that the entropy goes down and that is unnatural or entropically not favorable. But this loss of entropy can be compensated or recovered by the binding energy between the E and S, um, to form the transition state, because it can easily make the transition state. So you can have the that positive negative interactions, or polar to polar interaction, non-polar to non-polar interactions, and so on, which can lower the internal energy there, or the energy that is used for such reactions. And there are other advantages of using this proximity and orientation effects, like it gets rid of this solvent solvation shell, where other partner has to just swim through this solvent shell to interact with the actual side of the reaction there. And also there are negative charges there, and if it has to be oriented in a way that have to work with the other negative charged functional groups, it's basically there's a repulsion forces there, so it'll make it hard to interact with those molecules interact to each other, but the enzymes can help the interaction still, putting them at the proximity. And the last type of the catalysis is a preferential binding to ES complex, or the transition state. Again, this is important, and as the question, there was a question about this covalent bonding, if that is good or bad for the ES complex. But basically, I mean, this property, this type of covalent bond, solely uses the property where it forms the ES complex more easily to induce this catalytic reaction. And this enzyme basically can bind to the transition state intermediate instead of its original substrate in the original form. So it's going to have a much greater affinity for the intermediate rather than the substrate or the product, and that is really important property. But when you have a prolonged life of a transition state, meaning that if you have a much higher affinity to the transition state intermediates, that means you're going to have a higher chance of making ES forms, and this S becomes to the product, right? So ES concentration increases and higher chance to form the product. So it's a good thing, and that's the purpose of catalysis, basically. And how it does works is we can basically using some kind of like hypothetical scenario here, where we call the enzyme as a stickase. There's no such enzyme as a stickase in a real life form, but we just make an analogy here. And stickase, as it says, it breaks down the sticks, so that's why it's called a stickase. Again, this is a hypothetical enzyme. And let's suppose that its substrate is a metal stick here, and the reaction that has to be occurring is that substrate bends, and then eventually just broken into two pieces. That's the reaction that we want, and this is the energy profile that we can see basically from the substrate to product. So the product have a lower G value, meaning that delta G is it positive or negative? Is delta G positive or negative? I mean, here's a G for the initial, here's a final. Negative. So what does that mean? Does that mean this is a spontaneous reaction or not? Spontaneous. Again, if this process is not spontaneous, then even if there are good, enzymes available, it's not gonna, make it work, because enzymes won't change delta G, right? So this original reaction must have negative delta G value. So this is pretty typical kind of, energy flows. And let's say that there's one enzyme that can tightly bind to only the substrate. So let's say that this active site has lots of magnets, so you can hold this metal stick really tightly. But here the problem is that there's so much tight binding between the original form of substrate and the enzymes. It'll be much more stable in terms of this energy flow. So the ES complex supposedly here is gonna will have a much lower G value than S. So instead of going up, it'll be go down, meaning that your delta G double dagger, which is on free energy of activation, is going to be much higher to the compared to the uncatalyzed one, the original form. So basically, there's a huge dip in energy, so it will just stay here forever as a ES form, which is a bad thing, right? So instead of having this, the preferential binding to ES complex would be binding to this transition state, which is a bent stick. So let's say that your enzyme looks like this, and your active cleft looking like the resembles in this transition state. Then the original form, which is a straight stick, will get in there and will be bent naturally because of their magnets there, right? So it induces this ES transition state complex or the form. And now it can, basically lowers down this delta G double dagger again, which is a free energy of activation. It is now this is much smaller than uncatalyzed one, so it can easily just jump through this hurdle and gets to the final product, which is a broken to pieces. Make sense? Okay. And there are quite a bit of transition state analogs that uses this property where enzymes can bind to intermediates. And this is particularly really useful compounds for the drug design or the inhibiting something or boosting some kind of reactions too. So many enzyme inhibitors are based on this binding of the transition state intermediates, but without the true substrate intermediate, but with the something mimicking their structures. So again, it just resembles the transition state of the reaction. And transition state analog or TSA have these properties where it binds to the enzyme's active site tightly because it resembles the transition state where all the enzymes using this preferential binding to transition state catalysis. But this TSA can act as a potent inhibitor because they're not going to be processed and will nor make the final product. It's going to be just stuck there on the active site and will not do anything, just sitting there, right? So it's residing there. So it's inhibiting the interaction between the enzyme and the true substrates. That's why it's working as an inhibitor. And helps the confirming the structure of the natural transition state because it's going to be much more stable as opposed to the original transition state will be fastly just cleared to the product and the free enzyme form, right? But in this case, this TSA was stuck there, so you can actually examine the structures, how the active site is opened, and also interacting with those like analogs, which is mimicking the structure of the true substrate. And also you can actually use it on the different steps of the enzymatic reactions to tell the how those steps move through and the mechanisms are. And an example here would be the protein racemase reaction where the L form changes to D form. And this is the true transition state. But as you can see, these are the two transition state analogs, which is really mimicking this transition state of L-proline. But it has a little differences, like having double bonds or the missing some critical like atoms and so on. All right, so to truly understand how the catalysis happens, we need to take into consideration of the energetic changes, the energies, where the delta G is important, and delta G talks about the spontaneity, and that's all, right? But delta G double dagger, which is free energy of activation tells you that how much of this getting to the transition state gotten easier, how the transition the state energy goes down after the catalysis using the enzymes or catalysts. So the delta G double dagger tells you that the overall activation energy, and that has to be lowered during catalysis, basically lowering the bar for hurdle for the get to the product. And this delta G, since this is Gibbs free energy, it'll use the same equation where delta H double dagger minus T delta S double dagger determines the delta G double dagger. And delta H double dagger, as you know, is its energy, right? So the energy required for this reaction will be dramatically lowered if the delta G is lowered too. And vice versa, if delta H is lower, then that means delta G goes, decreases too. And also delta S2, it has to be increased, right? Because there's a minus here. If it is delta S double dagger is increased, that means you're gonna have a smaller delta G double dagger value. But when you think about the enzymes again, it's actually losing the entropy because everything has to be held into the active site. So you're actually losing a little bit here. So, but this loss that due to the substrate enzyme binding or the distortion of substrate or the restrainment, uh, some kind of like the limiting the motions of the substrate or the enzyme can be compensated by the a lot of like reduction in this delta H double dagger. Okay. So so far we talked about these kind of different types of catalysis, just a checking time, as well as the energy that is involved there. It's all about the lowering the delta G double dagger, right, which is free energy of activation. And now we're gonna keep shift the gear to the understanding how the all the those enzymatic or the catalytic steps work, and we can actually study the kinetics of that, putting the time value there. So we're looking at the dynamics of the molecules, how they interact, to understand more about their mechanisms. So why do we study the kinetics, enzymatic kinetics, or the kinetics for the catalysis? Because we can understand the enzyme properties, of course, as well as their mechanisms. And mechanisms basically elucidate the each step that takes place along the reaction pathway, so we can better understand how the active site works and how they select the specific substrates and things like that. And also we can understand how the substrate and enzymes binding, what kind of forces it uses, or the strength and stoichiometry, and so on. And also we can understand the how these enzymes working with the reacts to the activators and inhibitors, which can be used for developing drugs for the certain disease, and that is due to the mutation of the certain enzyme or the some chemical reactions that is over processing. So let's talk about the chemical kinetics. Again, this is a review, so I'm gonna go a little quickly, in here. If you don't understand, just let me know. So the chemical kinetics basically can be applied to the enzymatic kinetics too. And let's consider the simple elementary reaction where A becomes P. You can actually take it as a sequence of multiple elementary steps where A becomes one intermediate to the other intermediate and P2. And all these process, the rate of the reaction would be the summation of all the processes, but will be mainly determined by the slowest step. So that slowest step, it can be the A to I1 or I2 to P or so, will be called rate determining step. And that is mostly important step that you want to optimize for, drug design and so on, and also try to understand the how this enzyme works. And the for this A to P reaction, the rate of reaction or the velocity written as V here is described in the loss of substrate or the reactants basically over time, or the gain of the product molecules over time, and that's a concentration of the either product or the A molecule, right? So that means the concentration of A or P over time shows this rate of reaction and determine that. And we're going to quickly talk about rate law, where this we already talked about this velocity or the rate of the reaction, that is the changes of the A molecule or the substrate over time, and that is the same as K product of K and A concentration, where K is the rate constant that we talked about last lecture, basically. And when we talk about this, just A becomes P, that's a unimolecular reaction. But also there are reactions where two molecules are involved at the same time. So A and B collide to each other to make the C and D, in which case in this V, the rate of reaction will be K, again, rate constant, and times A concentration and B concentration. And this K is different from this K, right? Because it considers two molecules and their collisions at the same time. So that'll be called second order rate equation, or the second order rate constant for this K. And this is basically a bimolecular reaction. And here we call, and just, we can make the general form here of the reaction where A to Z, molecules all together just react to each other. And during that reaction, there are a fraction of a molecule, little b of the big B molecules, and so on. So that these coefficients can be also considered, because there has to be certain number of a molecule and so on. So they're gonna be put into the exponents here in the making into the exponential function. So again, coefficient in the general form will get to the exponent in the rate equation, or the rate constant. And let's quickly talk about the reaction order, because that is directly related to the exponents that we talked about, which is a coefficient in the equation. So in the reaction for the reaction rate V equals K, and then A concentration and B concentration to the zeroth, meaning that this B substance does not change over time during the course of this reaction, right? So it's basically can be involved there, but will not change in terms of its concentration, or might not be involved at all there. So this reaction will be first order reaction in terms of A, because there's a one A molecule involved there, but zeroth order for the B molecule, so that means independent from B. But the overall reaction will be the first order reaction, right? Because A one molecule and B zero molecule. So that's going to be the addition of these exponents, basically. We can apply the same thing for this bimolecular reaction where A molecule and B molecules are involved at the same time. Then it's going to be first order with respect to A alone or B alone. But overall, that's going to be second order reaction. Again, 1 plus 1, right? Right here. What about the overall reaction order for the V equals K A to the second and B? Yes? So it can be both ways. So one scenario can be where the B molecule isn't involved there at all, but it was just written there, well, by chance. And the other way is that it is involved there, but it doesn't actually do anything in terms of this reaction. It just have to be there, like solvent, or anything like any like environment that has to give there. So, but it does not involve in actual reaction. So exactly something like an enzyme. You can take it as an enzyme too. So again, what would be the reaction order for this V for A, two molecules of A and B, single molecule of B? Exactly, third order, because that's going to be 2 plus 1, right? It's the sum of the all the exponents, basically. So because the order, this order of the reaction corresponds to the molecularity of the reaction, how many molecules are colliding at the same time to occur this reaction. So obviously here there are 3 molecules involved there, right? And A and P is the first order, A plus B, P plus Q is the second order, and so on again. And mostly the chemical reactions are the zeroth, first or second, and rarely trimolecular, because it's really hard to even imagine that 3 molecules are involved at the same time making something, right? It's typically second order, up to second order. And also tetramolecular reactions are not known. So if you find it, that'll be a Nobel Prize finding. And this this enzymatic reactions basically have a substrate saturation effect, and that means we just talked about the reaction rate and reaction order and so on. That can be applied to the any kinds of kind of normal enzymatic reaction, but when you take into account of time points there or the concentration of substrates there, that actually there's some changes over time or with respect to the concentration of the substrates. So let's look at this curve where it shows the relationship between the substrate concentration and the velocity, the reaction rate. Of course, as you put more and more substrate, you're going to get higher and higher speed for the reaction, but it actually goes tapering down a little bit and makes this saturation point, because this is due to the number of enzymes that are available there, or the active site that can hold the enzymes at a given time, the turnover rate and things like that. So it'll make this kind of plateau here, which is called substrate saturation effect. But at the beginning, it's definitely a straight line, where there's a simple correlation between the concentration of substrate and the reaction rate. But overall, when you look at it, this is more like a hyperbolic. But we're going to mostly consider this just the initial state where the substrate concentration is really low, or it's just the beginning of the reaction, where we don't need to consider this hyperbolic effect or some kind of backward reaction or the reverse reaction too. If you have too much of substrate, then definitely there are going to be lots of products, and then there are going to be the reverse reactions accelerated too, right? So there are lots of things to consider. So to avoid that, we're mostly, during this course, we're going to only think about the initial reaction rate. So let's think about this and discuss this enzyme catalyzed reaction in more detail. We already talked about this substrate saturation effect, or the curve too. So basically, this is called substrate saturation curve, because you can see that how substrate concentration affects the saturation in the rate of the reaction. And again, this V is going to be proportional to the substrate concentration, especially at the beginning, where the concentration the substrate concentration is low. And since this is a simple correlation, it's going to be considered as just a first order reaction. But as concentration increases, now you're going to reach this saturation point. And that saturation point is indicated as a Vmax. So it's going to reach the Vmax and will stay there basically, as long as it's the substrate is provided, as well as the active enzymes. And At the high substitution point, the enzyme will, all of the enzyme will bound to S to make the ES complex. And that can be basically resolved, or going back to the free enzyme form when the product is formed, or in which case another substrate can be bound to the enzyme right away, or this substrate, instead of becoming to the product, it can go just go back to the substrate from the ES complex, um, depending on the probability, at the top of the transition state. In which case, again, another new substrate can still jump on the enzyme to form the ES complex. And this discovery of the substrate saturation effect and the drawing the curve is really important. It's a cornerstone of enzymatic studies, especially kinetics, because it literally suggests for the first time that there's a direct interaction of the enzyme and substrate to make the ES complex. Otherwise, you couldn't really explain how enzyme worked. So here's basically a simple diagram where E and S forms the ES complex and then becomes to the product and the free form of the enzyme. And basically you can get rate constant of all these reactions. So E plus S to ES will have the rate constant of K1, and the reverse reaction will be K minus 1. Same thing applies to this ES to E plus P will be K2 with the K2. So you can say that the reverse reaction from E plus P to ES will be K minus 2, right? But at the beginning of the reaction, there only a little of a substrate, so all of the substrate will be processed to become the product really in a fast rate. So basically, you're not going to have much of a K minus 2 or the E plus P to ES going back. So we're going to ignore this K minus 2, which again make it much more simple. But it's only applied for the initial reaction steps. So Michaelis and Manton suggested a theory in 1913 to explain how these enzymatic reactions happen. And what they did was that basically they suppose that there's an ES complex because of this substrate saturation effect. And that was served as a foundation of a modern enzyme kinetics. They have the really important two simplifications or assumptions here. First, they assume that there's an equilibrium between binding E plus S to become the ES, and also ES dissipates back to the E plus S. So equilibrium between these reactions. And also they assumed that for another simplification, that this K2 that gets to the product is much slower than this K1 or K minus 1 reaction. Does it make sense? That so here there's a equilibrium, so it'll go back and forth really quickly, as opposed to ES complex will just rarely goes to the E plus P. And that those are the assumptions. But as you can actually feel that, that's not all, always happen, because I mean many times you have lots of products immediately, right? And it seems like these assumptions basically tell that there's a bottleneck on the ES, and there are a lot of things going back and forth between this substrate and the ES complex. But it's not all the cases. There are many cases just the ES becomes P quite a lot. But again, this is a really important theory to understand how this enzyme works with the substrate. If the rate of the formation of the ES complex is the same as rate of the breakup, E plus S, then Ks value, which is a substrate constant, will be written as basically E concentration, S concentration over ES concentration, which is the same as Kd, right? Dissociation constant for ES complex. And if you plug the velocity, the rate reaction equation, where V equals ES concentration with the K value, you can put it there. The ES to the E plus S to ES will have K1, and ES will have K minus 1, and it has to be flipped because the V equals K1, E concentration and S concentration. So you need to plug the velocity there. And velocity is the same because the rate is the same here. So they're going to be canceled out. I didn't write it here, but basically that's going to be the ratio of K minus 1 and K1. And this, like I said, it's going to be same as dissociation constant. So let's see. After that, Michaelis and Menten theory is developed after like 13 years or so. After that, they found that there's some problems there where it does not actually cannot be applied to all the enzymatic reactions. Again, those assumptions are too extreme, so they cannot be applied to all those reactions. So Briggs and Haldane added more ideas to make it more applicable to real enzymatic reactions. So what they did was that instead of having the equilibrium state between the E and S complex and E and S molecules and the ES complex, they actually suggested that the ES complex will be at the steady state. So they didn't say anything about this K2 is much smaller than K1 or K minus 1, or this rate is much slower than this rate. What they said was that they're going to be a steady state. It's different from equilibrium state, but there are going to be a lot of input here, but a lot of output here at the same time. We'll have the steady state in terms of this ES concentration. And that totally makes sense, right? Because your enzymatic reaction has to be like keep going, and then should be quite fast to produce the product. And this ES concentration will be the constant value, meaning that the ES, when ES is formed, it's going to be dissipate into the back to the E plus S form, or goes to the E plus P form to make this enzyme available and interact with the S again. And again, this is called steady state assumption. And this assumption is actually validated as true because you can measure the concentration of product or the substrate using the apparatus called stop the flow apparatus. And you just stop the flow of substrate and measure the how much of product you gain. So basically, you can measure the speed of the rate of the reaction or the rate constant of the reaction experimentally here. And this assumption was proven true by having the predicted data and comparing with the experimental results or the measurements there. So this is the last slide. We just talked about the steady state assumption. This is the key of this again right here, Briggs and Haldane hypothesis, where they said that these simplification of Michaelis and Menten theory is too extreme or too restrictive. And that was all about the ES complex has the steady state. So let's think about the enzymatic reaction here over time. And you're looking at the concentration of enzymes, substrates, as well as products too. So of course, when you look at the substrate, it's going to go down over time, right? Because it's going to be processed to the product. So the product concentration will go up. And when you look at the total concentration of enzyme, it must be the same, right? Because it's not going to be involved in any reactions. So it's not going to be dissipated. It will stay there forever. Not forever, but for the time of these reactions. But free enzyme will go down as opposed to ES complex will go up. And once it reaches to the, some kind of maximum point, then it will stay there where the ES complex concentration change is zero, right? That's what steady state assumption is. And that's not about the equilibrium. It does not have to reach the equilibrium, but it's just fast enough to produce a product. So it'll have that kind of plateau in terms of making their ES complex. And then we'll go down eventually when all the substrates are processed there. And you can zoom in right here at the beginning of the reaction where everything is behaving something like the more like linear, where the ES complex goes up and also the product goes up. And again, this is the initial state that we're going to consider in the most cases. That's it. And again, when you have questions, just to come to the office hour or email me.